All right, welcome back to the last installment of the steps of a muscle contraction. So today we're going to be talking about the Crossbridge cycle. So in the Crossbridge cycle, let's preface kind of where we were in our previous lectures here, where we talked about the excitation or the events at the neuromuscular junction, the excitation contraction coupling, and then we'll jump into the structures as it relates to the cross-bridge cycling. So we first started off with this idea of an action potential. The action potential starts at the sarcolemma, but it's stimulated by a release of acetylcholine into the synaptic cleft. So the neuron here, as you can see on the top of the screen, the neuron's going to release acetylcholine into the there it is, acetylcholine into the synaptic cleft that'll bind to receptors located on the motor end plate of the sarcolemma. When the receptors are initiated, these are ligand gated ionotropic receptors that become permeable to ions. So sodium, potassium, calcium can freely pass through pass through these ionotropic channels once they're open. So it's the influx of sodium that allows for the action potential, the upset of the membrane potential, and that travels across the entire sarcolemma. From the sarcolemma, it travels into the T tubules, right? So it travels into the T tubules and into the and that the stimulation then stimulates the sarcoplasmic reticulum. Let's take a look here in this slide as we were talking here. First, it's the stimulation of the nerve, which causes an action potential. The action potential will travel That action potential will travel across the entire sarcolemma down into the T tubules, and then it will stimulate the sarcoplasmic reticulum to release calcium. Once calcium is released, uh, remember that there is the two regulatory proteins on troponin, on actin, the troponin and tropomyosin. There is the myosin here in the middle. So once calcium's released, this is where we get the actions of the cross bridge cycle. So what we have is in this picture, we have the actin molecules in green. You've got the long strand of tropomyosin in yellow here, and the tri-complex proteins of troponin. So when calcium binds to troponin, it causes a shape change. These actin molecules are allowed to bind with myosin, and the first thing that we get is a cross-bridge attachment. So all the way back from when calcium is released from the sarcoplasmic reticulum, they bind to this structure here. It'll then cause a change in shape of the tropomyosin, allowing actin to bind to myosin. Now, the next thing that happens is the power stroke, because as you'll see in the next phase, the energy is used to detach. You can see there that the energy is then used to detach and then put the myosin in the high energy position, or in the cocked position, as it might be called sometimes. So again, the phases of the cross bridge cycle, there's four of them, as we can see here. You've got cross bridge attachment that becomes passive. So there's the attachment, the releases the ADP and the inorganic phosphate, but there's the power stroke, cross bridge detachment, and repositioning. So cross bridge cycle, again, is the four major steps that occur at the end of a muscle contraction when they're actually generating force, right? So this is actually when force is being generated. It starts again with the tensions being generated, right? It starts when the cross bridge actually attaches. So you've got cross bridge attachment, power stroke, cross bridge detachment, and then finally the repositioning of the myosin head. And so this will happen repeatedly as long as there is calcium being released. So calcium stores within the cytoplasm to maintain the tropomyosin staying away and allowing the two binding sites to be attached, as well as long as there is enough energy for this to go on. And so I'll finish it off by demonstrating here with a animation where we get to see the Crossbridge cycle in action. So now, as you can see here, we're going to focus into the skeletal muscle, and we'll look at the Crossbridge cycle specifically. So recall that there's the neuron there that stimulates the muscle fiber. The structure of that is the sarcomere, the functional unit of a muscle contraction. Sarcomere is made up of alternating thick and thin filaments. Now, the stages of a crossbridge cycle, once calcium is released into the sarcoplasm, it's going to bind to troponin. It's going to cause tropomyosin to move out of the way, revealing the binding sites for myosin. There we go. Binding sites are activated. And the first thing that's going to happen is attachment. So once you've got the myosin head binding, that's the first step. Myosin head binding to actin. The next step is that ATP will detach and then reposition. We didn't get the power stroke. We didn't say detach and reposition. So let's go take a look at this crossbridge cycling and all of its steps. Step 1, crossbridge formation, attachment, crossbridge attachment, bang. The second step is power stroke. Here it is, power stroke. It's passive. It allows this to become, the power stroke is passive. You don't need energy initially to occur the power stroke. And then the next is that ATP will bind to the myosin head, causing it to detach, and then reposition or place it in the cock position. So there's detachment, and then reactivation or repositioning or cocking of the myosin head. So as energy is taken in winding it up, it's almost like a wind-up doll. You're using the energy to wind up the myosin head, and then once you're in that position again, you're going to power stroke. So as long as there's binding sites on myosin and actin are revealed, and as long as there is enough energy, ATP, to continue this, muscle contractions will continue. And what the ultimate concept here is referred to as sliding filament model of a muscle contraction. And so there we have the major steps of a muscle contraction. We've seen these in the previous section as well. The excitation, events that occur at the neuromuscular junction, excitation-contraction coupling, and now we finished here with cross-bridge cycle.